Paul Thomas, Senior Editor of Pharmaceutical Manufacturing Magazine and PharmaManufacturing.com. I'm at Interfex 2009 and I'm talking today with Cindy Lieberman. And Cindy is the Vice President of Marketing for Celsis. Cindy, thanks for being with us today.
My pleasure.
And Celsis does a number of different things, so maybe you could start out by telling us what Celsis does and what your areas are.
I'd be happy to. Celsis has 3 business units. At different ends of the manufacturing spectrum, we have an in vitro technologies group that focuses on early stage drug discovery and development, preclinical work. Our analytical services group, which handles chemistry, microbiology, and stability testing programs across from the early stage clinical trials all the way through finished products. And then our rapid detection division, which companies that are manufacturing use it to test raw materials, work in process, and finished products before they're released to market, make sure they're free from microbial contamination.
Well, let's start out talking a little bit about your analytical services. What are you seeing out there? Why are people coming to Celsis for help?
Well, we're very knowledgeable about the compendial methods, both in the US as well as in Europe and Japan. And we've helped a lot of our customers prepare for changes to the USP. That's a very hot topic. Recently, in July 1st, was the deadline for residual solvents. And we did a lot of work in our chemistry department helping customers make sure they had the protocols in place and were able to test for that. And even some that have missed the deadline that are still trying to catch up. And now there's a new deadline looming on May 1st for microbial limits testing. It's changes to the USP Chapter 61 and a new Chapter 62. It's part of the USP being harmonized with the EP and the JP, the European Pharmacopoeia and the one in Japan. So there's a lot of work now to help customers get prepared and revalidate their products for the new chapters.
So if a customer came to you and said that they're concerned about this May 1st deadline, for example, what kinds of things would you provide for them?
Well, the first thing is to get started as soon as you can, because it could take, depending on what the dilution tests show, it could take up to 28 days to validate your products. Hopefully it can be done within a week if you have a good sense of what growth media needs to be used, but if that dilution's not right the first time, you need to try it again from 10-in-1 to 100-in-1 to 1,000-in-1, and that can take time, and May 1st is really just around the corner if it's going to take that long. So we have some resources on our website at celsis.com/USP that talk about the changes coming, and also some FAQs about what people should think about in terms of sample sizes they should submit and what to expect and the kind of information we need to help them through this process.
Good. Let's talk also about rapid detection. What's going on with Celsis in that area right now?
That's a very busy area now. Probably the number one thing we're seeing is companies that have been using us for sterility testing now, or an interest in getting validated to use it, rapid detection for sterility testing. The typical sterility test is, as you know, like 14 days or more. And the research we've done, in fact, we recently published a poster, shows that you could use the Celsis Rapid method equivalently in 5 to 6 days, 5 to 7 days. That's a huge savings. It cuts the time in half.
So what kind of value does that provide to the customer then?
Well, when a customer can go from holding products for 14 or more days to only 7 days or less, they've freed up their inventory requirements, freed up warehouse space, get products to market faster, and all that has a result of freeing up working capital.
I also understand that you're introducing something new here at Interfex in terms of rapid detection as well.
That's right. We have a new product called React, and it's the first fast and easy to use RNA-based assay that gives you results within 2 hours. So if you're using our primary screen, 95 to 98% of the time you're going to have clean results and you get your products to market. But for that 1 to 5% of the time where there is a Celsis positive, as our customers say, you could use the Celsis React to identify if it's a gram-negative, if it's Staph aureus, or if it's a yeast or mold, or if you have a particular wild strain that you're concerned about or an organism that keeps coming up in your plant again, we can talk about creating a special probe just for your use.
So you would see this as a complementary product to what you have already on the market then?
Definitely. It follows our AccuScreen test, which provides results within 24 hours, including for absence of mold. And if there is a positive, then within 2 hours after the first screen, you will know if it's a gram-negative or another objectionable in your product.
Yeah. And clearly there's a benefit for discovering contaminants in your processes, things like that. But how do you help your customers to really discover the value of your product and the value to them, and making sure that everything is clean and in working order?
That's a great question, because the value is really beyond the time savings in the quality lab. Often the value of implementing a Celsis rapid detection system is felt company-wide. So we created with one of our customers early on, and the Arthur J. Little company of management consultants, what we call the value creation model. And we'll sit with a company that's interested in the method, and there are about 20 different data points that we can enter into this program. It includes things like what the average lab tech costs, what the average cost of the current sterility or current testing method is, how much the daily output is, how many tests are run a year, and those data points combined into this value creation model will give you an ROI, which is usually somewhere around 6 to 9 months for micro limits and less for sterility, and a net present value, which is a 5-year, how much free working capital we're freeing up. The average for micro limits about 500,000, and it could be $1 million or more for sterility testing.
That's a nice tool to have, I would imagine.
And we've been using it for years. The data is very accurate. Our customers tell us once they've implemented that is in fact what they're finding.
Great, and there are a lot of customers that are very familiar with rapid microbial methods, that really have a handle on what they need to do, but then I feel like the industry is still kind of getting up to speed. What's your take on that? Where is the industry in terms of rapid micro? Where does it need to be?
I think that the time has come, particularly in the pharma area. The FDA's been behind rapid detection, rapid methods for a while now, and we do have customers that are having it validated for sterility, which I think is the next big time savings. I like to think of rapid detection as one of the last great frontiers for huge process savings in the manufacturing cycle. Everyone's leaning in different areas, and this is a huge opportunity to really get the company leaner, because you're taking days out of the cycle time, not just 20-minute savings here by rerouting a line. Days are out of your cycle time. It's a tremendous opportunity for companies to get leaner and to be more efficient.
Yeah, it sounds like it. Well, Cindy Lieberman, thanks so much for talking with us today.
My pleasure.
Really appreciate it.
Thank you.